Hi everyone. Today we will learn about chapter 3, the basic principle of heredity. First, we will learn about Mendel, and our second, we will learn about monohybrid crosses. Third, we will learn about the dihybrid crosses, and also fourth, we will learn about uh, the statistics, some statistics. Okay, first, okay, in this slide. So this is the blonde hair, which is observed in the Solomon Island. So this is Australia, and this is New Guinea, and we call it as this is Solomon Islands. And also in Solomon Island, blonde hair occurs in 5 or 10% of dark-skinned Solomon Islands. So his skin color is dark color, but he has the blonde hair. But we found that this blonde hair in this group is genetically different from the blonde hair in European. This is blonde hair in European peoples, and so finally people found that this blonde hair is a recessive trait. This is due to a point mutation in the TYRP1 gene, and specifically the 483 cytosine is changed to threonine. So this is the TYRP1 gene. So around this location, only one nucleotide C is changed to T, so it can induce blonde hair. But this is the recessive trait, and this blonde hair is different from the genetic basis from or different genetic basis from blonde hair in the European. So in this slide, we will learn about Mendel. So actually, the Mendel discovered the basic principle of heredity. So actually, he was the Catholic priest, but he discovered the basic principle of heredity. How he can discover the basic principle of heredity? Because he used the proper experimental model, and also he used an experimental approach, and you analyze the result mathematically, and also he studied easily differentiated characteristics. Okay, let's start from first. So he used the proper experimental model. So he used P. In Korean, we call it as Wandu, because Wandu has the P has a short generation time, and also at one time it can produce numerous progeny, and also P has the ability to carry out controlled genetic crosses. And also he used an experimental approach like the controlled genetic crosses, and also he analyzed the result mathematically, because the P can produce numbers of progeny, maybe at one time more than 1,000 or more than 100, so he can use the mathematics. So he analyzed result mathematics. And he also studied easily differentiated characteristics. He used the 7 characteristics that appeared in the seed and plant. For example, first one is seed color, yellow or green. Second one is seed shape, round or wrinkle. The final one is stem length, short or tall, like this. So because of these 3 criteria, he can discover the basic principle of heredity. Okay, this is concept check. Which of the following factors did not contribute to Mendel's success in study of heredity? A. His use of pre-plant. B is his study of plant chromosome. C is his adoption of an experimental approach. And D is his use of mathematics. Did not contribute to Mendel's success? The answer is B, isn't it? So he did not study about plant chromosome. He studied 7 characteristics like this, okay? In this slide, we will summarize important genetic terms. For example, start from gene, allele, genotype, or phenotype or trait, or characteristics. So what is gene? In Korean, we call it as 유전자. So gene is in English, we call it as an inherited factor that determine a characteristics. 한국말로 특징을 결정하는 유전 요소가 되겠습니다. 아니면 DNA지요. 그러니까 seed shape를 결정하는 유전 요소를 유전자라고 합니다. 다음에 allele. Allele is one of two or more alternative forms of gene. For example, seed shape. One is the round, one is wrinkled. So for example, we have the homologous chromosome. So this gene is the gene which involved in seed shape is located around here. So we call it as allele R, large R. So another allele, different allele, for example, this allele includes wrinkled seeds, so we call it as a small r. So different allele for a particular gene occupy the same locus on the homologous chromosome like this. So large R or small r. So allele means that one of 2 alternative forms of gene. For example, seed shape, large R or small r like this. So what is genotype? Genotype means the set of allele possessed by an individual organism. For example, in the peas, the round seed is the genotype will be large RR or large R, small r like this. So this is, we call it as genotype. Maybe wrinkled will be the small r like this. So small r will be another genotype. So what is phenotype? So phenotype is appearance of the characteristics. So phenotype means that round or wrinkled. We call it as phenotype. Phenotype, in Korean we call it as 표현형. 한 특징의 외적인 출현을 we call it as phenotype이라고 부르고 있습니다. 그래서 라운드나 wrinkled를 we call it as phenotype이라고 하고 있습니다. What is the characteristics? Characteristics is a feature possessed by an organism. 그러니까 characteristics이나 phenotype이나 아주 비슷한 말이죠. 특징 해가지고 색깔 또는 모양, 일반적인 현상을 의미하는데 phenotype or characteristics are a little bit similar. So for example, round shape will be the characteristics and wrinkled shape also will be the characteristics. Okay? So in this slide, we will study the monohybrid crosses. So actually the monohybrid crosses reveal the principle of segregation and the concept of dominance. Okay? So what is monohybrid crosses? Monohybrid crosses cross between two parents that differ in only single characteristics. Everything is the same. exactly same, but they only differ in the, for example, in these cases, they only differ in seed shape. For example, this is a round and this is wrinkled. So in these cases, we call it as monohybrid crosses. So for example, this is the this is the experimental question is when peas with two different traits, for example, round and wrinkled seed are crossed, will their progeny exhibit one of the two traits or both of those traits or on the intermediate trait? What is the answer? The answer will be the progeny exhibit only one of those traits. So we will see the details like this. For monohybrid crosses, for example, this is the anther and this is the stigma. This is stigma, for example, to prevent different variety of to cross different variety of fish. Mendel removes the anther from the flower to prevent self-fertilization. After crosses, he found that, so he crosses homozygous, he used homozygous round seed, and also he used homozygous wrinkled seed. And he did monohybrid crosses. Only one is different. For example, seed shape. So this is F1 generation. He found that all of the F1 seeds were round. And also he did the self-fertilization. He used the F1 generation. So he did self-fertilization. So he used F1 generation. So he did self-fertilization. And finally, he found that three out of four is a round shape, and one out of four is wrinkled shape. So based on this experiment, we can conclude like this. So F1 plants display only one phenotype of parents. For example, this one only shows the round shape. F1 generation is only round shape. And also both traits are passed to F2 generation in the 3-to-1 ratio. So why it can be passed to the 3-to-1 ratio? Because the two genetic factors, for example, large R, small r, are separate when gametes are formed. For example, large R, small r can be separated. So one can be separated large R, or one can be separated small r. So in the father, this is mother, and also this is father. Also this large R is separated large R, and the small r can be separated to small r, right, like this. So this is monohybrid crosses. So one character is encoded by two genetic factors. For example, seed shape is encoded by two genetic factors. For example, large R or small r. Homozygous round seeds will be the large RR. Homozygous wrinkled seeds will be small rr. And also two genetic factors, large R, small r, are separate when gametes are formed. So when 생식세포가 만들어질 때 large RR은 large R, small r로 separate 되고 생식세포가 만들어질 때 large R, small r은 large R, small r로 separate 된다. Okay, so through the monohybrid crosses, people found that Mendel found that the concept of dominant and recessive traits. For example, seed shape, what is dominant and what is recessive? Round is dominant and the wrinkled is recessive because the after crosses, F1 generation, because or even though their genotype is large R, small r, so F1 plant only shows the round shape. So it means that round is dominant over wrinkled. And also, second one is that the two alleles are separate with the equal probability to the gamete. For example, after that he did self-fertilization using the F1 generation. So finally, in F2 generation, he found that 3:1 ratio of the round to wrinkle. So this is due to the two alleles of the separate, two alleles can separate with equal probability, like this. Two alleles can be separated equal 1:1, 1:1 ratio, like this. Okay, so this is the Mendel's first law, principle of segregation. So what is principle of segregation? So first one is each individual organism possesses two alleles encoding a trait. For example, seed shape, large R or small r. And allele separates when gametes are formed. For example, this is anaphase 1. We already learned about meiosis. So during anaphase 1, homologous pairs of chromosomes can be separated into each pole. And also, allele can separate it in equal proportion, 1:1, 1:1. So it happened during the anaphase 1. So this is the Mendel's first law. Mendel's first law is principle of segregation. So each individual organism possesses two alleles, and also alleles separate when gametes are formed, and also alleles separate in equal proportion. So Mendel proposed the principle of segregation. So what was the principle of segregation? We can go back to the previous slide. The allele is separated when gametes are formed, and also allele separated in equal proportion. And finally, he got the 3:1 ratio. But at that time, he didn't know about why this kind of 3:1 ratio is happening. But times later, Thornton found that chromosome. So he proposed the chromosome theory of heredity. So after he proposed the chromosome theory of heredity, he can explain the principle of segregation very well. So what is chromosomal theory of heredity? So each homologous pair of chromosome, for example, the red one comes from maternal chromosome, and the blue one comes from one paternal chromosome. So these pairs of the chromosome segregate independently into the gamete 배우자 in the meiosis. After they experience the prophase 1, meiosis 1, during anaphase, each pairs of chromosome is segregated independently. So each pairs of chromosome segregate independently. After that, these pairs of chromosome, they made large R allele, and these pairs of chromosome, this chromosome, they can make small r and small r. So it means that so at that more after F1 fertilization, so we can get the 3 to 1 ratio like this. So this is the chromosomal theory of heredity to explain the principle of the Mendel's principle of segregation. So it means that the pairs of the homologous chromosome segregate independently in the anaphase 1. That's why they have the same ratio. They have a 2R, 2 large R, 2 small r. The ratio is 1 to 1, like this. Okay. Next, to explain the Mendel's principle of segregation again, we have to understand the Punnett scale. So if we use the Punnett scale, we can predict outcome of genetic crosses. What is Punnett scale? This is Punnett scale. So we will see. Okay. So this is the P. One has they are large, like this. One is the small. So, but this is the heterozygote, large T, small t, and the smaller one is the small t, like this. This is the large T is dominant than small t. So, if we use the Punnett scale, we can predict the phenotypes very well. For example, in F1 generation, for example, this gamete can make they have the large T, small t allele. So this gamete can make the large T, small t like this. How about the gamete from the smallest the P? Their genotype is small t, so we can write down small t like this. So in these cases, large T, small t can match each other. So genotype will be the large T, small t. In these cases, it's a large T, small t, and large T, small t like this. So this is the taller Phi, and this is tall. Okay, how about this? The scale. Small t and small t meet each other, they will make the small tt. So, and the phenotype will be the short. How about this scale? And small t, they met each other, so they will make the short one. So finally, the genotype ratio is large T, small t is 1, 2. Small t is 1, 2. The ratio is 1 to 1. How about the phenotype? Phenotype is also tall and short is 1 to 1. So we can predict the outcome of genetic crosses by using Punnett scales very well. Okay. So in this slide, we will solve the concept check 2. The question was the F1 plant is backcrossed to the parent with a round seed. So okay, the parent was one is they have the large RR, and means that the shape is the round. Another allele was the small RR, and shape was the wrinkles. So in the gamete 배우자에 they have in one 배우자 they have the large R, one 배우자 they will have the small r. So after fertilization, the F1 generation genotype will be the large R, small r. How about the phenotype? Phenotype will be the round like this. So it means that F1 plant, large R, small r, large R, small r is backcrossed to parents with a round seed. It means that the genotype will be the large R, So in Punnett scale, so we write down here large R, small r, and also write down here large R, large r. So if we use the Punnett scale, in each scale they will have large R, large R, large R, large R, and large R, small r, large R, small r. So the phenotype, all of them will be the round. So what is will have the wrinkled seed will be the 0. Okay. This slide we will learn about the probability. Probability means that the likelihood of the occurrence of a particular event. So in genetics, we usually use probability to predict the outcome of a genetic process. There are two rules. One is the multiplication rule, and another rule is the addition rule. Okay. You have a die 주사위 like this. So if you roll a die like this, what is the probability you will have to obtain 4? What is probability? Probability will be the 1 to 6, because the case is you will obtain the 4 in the upstairs will be the 1 out of 6. But if you roll the die again, what's the probability of getting 4 again? It means that it will be also 1 out of 6. So the probability of getting a 4 on first roll and the second roll again, it will be the 1 out of 6 multiply 1 out of 6, it will be 1 out of 36. So this is the multiplication rule. So what is the addition rule? Addition rules means that what is the probability getting either a 3 or 4? It means that the chance you will get the 3 is out of the is the 1 out of 6, and also you will have the 4 will be also 1 out of 6. So finally, the probability of getting either a 3 or 4 will be the 1 out of 6 plus 1 out of 6 will be finally 1 out of 3. So this is the addition rule. Okay. Multiplication rule means that there will be and addition rule there will be the or. Okay. So in this slide we will solve the concept check 3. So okay, if the probability of being blood type A is 1 out of A, and the probability of blood type O is 1 out of 2. So what is the probability being either blood type A or B? Either A or B. It means that this will be the addition rule. This will be the either A or B. It means that this will be the addition rule. So it means that the probability of being blood type A is 1 out of A, and the blood type O is 1 out of 2. So finally, it will be the 5 out of 8, like this. Okay. So in this slide, I will explain the binomial expansion. In Korean, we call it as 이항정리. This binomial expansion is very useful for complex situation. The binomial expansion takes the form of P plus Q and like this. What is P? P equals the probability of the one event. The other Q means that Q equals the probability of the alternative event. What is n? N is equals the number of times the events occur. So okay, so you cannot understand the concept of binomial expansion based on this explanation. So we will solve the one problem. Okay. So the mom and the this is the albino. Albino is the genotype is must be the small a and small a. Okay, like this. This is albino. Okay. So mom and dad are both heterozygous. So mom is large A, small a, and dad is also large A, small a. And also because large A is dominant than small a, the parents have normal pigmentation. But they still have the carrier like this, small a, but after the parent, after mother and father crosses, the probability of child having albinism will be 1 out of 4, isn't it? So because based on this Punnett scales, this will be the large A, large L. So it means that this will be the normal, and this will be the large A, small a will be also the normal pigmentation. Either large A, small a also be normal pigmentation. But in small a, in these cases, the child will have the albinism. So the probability P equals probability of one event. One event means that albinism. Albinism will be the 1 out of 4. Q means that this equals the probability of the alternative event. Q means the probability of the child having normal pigmentation, large AA or large A, small a. It will be the 3 out of 4. So Q means that 3 out of 4. So what is N? N means that N equals the number of times the event occur. Okay, let's explain in next slide. Okay. What the probability is of this couple having 3 children? This couple is they are heterozygous like this, large A, small a, large A, small a. In this case is having 3 children, 1 with albinism and 2 with normal pigmentation. So it means that 3 children, 3 will be the N. So N equals number of times the event occurred. Okay, the number of events will be this couple have the 3 children, 1 with albinism and 2 with normal pigmentation. Okay, 1 with albinism and 2 with normal pigmentation. So okay. In one case is let's explain. One case is albinism. Second case is will be the normal like this. Another case is the first child will be the normal. Second child will be the albinism, and third will be the normal. Another third case is the first child will be, second child will be normal, and third child will be the albinism. So the probability is, this is the multiplication rule. So 1 out of 4 multiply 3 out of 4 and 3 out of 4 like this. This is also same like this. So it means that the probability will be the P multiply, P will be the 1 out of 4. Q means that 3 out of 4 multiply 2 like this. So, okay, what is the probability of this couple having 3 children, 1 with albinism and 2 with normal pigmentation? the probability so we have to this will be this case either 1 or either 2 or either 3. It means that we have to apply addition rule. So it will be the 3p multiply q square like this. So the probability will be like this. Okay. So this case is we can solve it like this, but we did not use the binomial expansion. But if we use the binomial expansion, it will be more simple. Okay. So this is binomial expansion means that p plus q multiply n like this. So n means that for n means that they will have 3 children. So n will be the 3 like this. So if there will be the p plus q, then this is 3 will be like this. It will be if there is 1, it will be p plus q. If it will be 2, it will be the p2 plus the 2pq plus q or q square like this. If the factorial n means the n will be the 3 children, it will be the 3 and they will have like this. So if you use the binomial expansion, so the probability of this couple having 3 children, 1 with albinism and 2 with normal pigmentation. It means that we can collect so the n will be the 3. So it will be the 1 3 3 1. So but the probability will be 1 with the albinism, so they will have only 1 p. Another 2 children will be the normal pigmentation, will be the q square like this. So the probability with 3 p q square like this. So we can solve the problem is like this by using the binomial expansion. Okay, so this is already difficult. So next solution will be like this. So we can use this one. So binomial expansion is very difficult to understand. So if you use the Pascal's triangle, so it will be a little bit simple. But if you use this method, it will be very simple. Okay. So it means that the m means that the overall probability of the event x, the albinism with the probability with the p. P means that 1 out of 4 occurring. How many times occurring? Occurring 1 times. How about event y? Means that normal with the probability will be the 3 out of 4. It occurs 2 times. So what is the probability of this couple having the 3 children, 1 with albinism and 2 with normal pigmentation? So it will be n will be s plus t. So the equation is like this. P is the n means that 1 plus 2. So it will be the 3 factorial. And the s will be here 1 factorial. And t will be here 2 factorial. And p and s times means that it will be 1, and q t times means the squares like this. So finally, it is, the equation will be p is the 3 p q squares like this, and the probability will be like this. We can solve it like this. Okay, so if you use this square, it will be very simple. So this is also another example of binomial expansion. So in this slide, we can solve one problem. Okay, what's the question? What's the probability of this couple having 5 children? N is 5 children, and 2 with albinism and 3 with normal pigmentation. So based on the coefficient and terms or for the binomial expansion, P plus Q, the N, is very difficult to predict, but based on this, the equation, it will be very simple. So 5 will be the 5 factorial, 2 with albinism, 2 factorial, and 3 with normal pigmentation, 3 factorial. At that, the 2 with albinism, so 1 out of 4 square, and 3 with normal pigmentation, 3 out of 4, 3. So finally, the equation, the probability will be the 0.26, like this. So in this slide, we will learn about monohybrid test crosses. What is monohybrid test crosses? It means that, for example, large T enclosed tall height and small t enclosed the short height. But for example, this P shows that the phenotype is the taller and taller, but we don't know the genotype because they can be the large T or large T small t. So it means that one individual of unknown genotype is crossed with another individual with a homozygous recessive genotype. Homozygous recessive small tt. In these cases, this is Punnett scale. For example, this is Punnett scale in F1 generation. This is a large t, small t, small t, small t. So in this scale will be the large t, small t. The phenotype will be the tall height. How about this one? This scale will also be large t, small t. The phenotype will be the tall height. How about in these cases? The Punnett scale shows that small t, and the height will be the short. And how about this case? The phenotype will be also short. So genotype, the ratio between the large t, small tt is a 1-to-1 ratio, and phenotype is also 1-to-1. So one individual of the heterozygotes, the conclusion will be the 1-to-1 and 1-to-1. How about if the one individual unknown genotype is large TT? In this case, this is Punnett scale. The large TT can go to the T, large T, and small TT can go to the here and there. So after Punnett scales, so this scale will be the large T, small t. This one is also large T, small t. This one is also large T, small t. This one is also large t, small t, and also genotype ratio is 4-to-0, and also phenotype ratio will be the 4-to-0. So if the phenotype shows 4-to-0, it means that one individuals of unknown genotype will be the large t, large tt. Okay, so this is through by using the monohybrid test crosses. We can know that one individuals of unknown genotype. So based on this based on this analysis, so we can know that this is a large t, small t, heterozygote or the homozygous, like this. Okay, this slide we will cover the dihybrid crosses. What is dihybrid cross? Dihybrid cross means that examine the two traits at a time. Previously, we covered monohybrid crosses. What was monohybrid cross? At that time, we only examined one trait at a time. At that time, we only cover one trait, for example, round, the wrinkle. We only cover the shape, or we only cover the color, yellow or green. So what dihybrid crosses examine two traits, for example, shape, round and wrinkle, and the color, yellow and green. So the question was that do alleles encoding different traits, for example, the allele, the large R encoding rounds, and also large Y encoding yellow color, separate independently? To answer this question, Mendel established homozygous dominant genotype, something like the large R, large Y, large Y. He also established homozygous recessive trait, small r, small y, small y's. So the phenotype was the round and yellow seed, and the wrinkle and green seed. So from this genotype, the gamete will be the large R, large Y, and from this phenotype, the gamete will be large, small r, small y. After that, these two gametes will be will do fertilized, and this is the F1 generation. F1 generation showed that round and yellow seed, because round is dominant than wrinkle, and yellow is the dominant than the green. So this is the F1 generation. So he did self-fertilization. And finally, he found that the ratio 9 to 3, 9 to 3, to 3 to 1. 9, 3, 3, 1 means that 9 means that yellow and the round and yellow, and also 3 means that 3 means that round both are green, and 3 means that in the wrinkle but yellow. This wrinkle but yellow is 3, and 1 is that wrinkle and greens like this. So, but what's the answer? Do alleles encoding different traits separate independently? For example, if the allele encoding the shape and the color is separate independently, the gamete will have the large R, large Y, and at that small r, small y, and another one is a large R, small y, another one is that small r, large y. So if the gamete two traits is separated independently, the gamete will have this kind of the genotype. So how about we will do the Punnett scale? This is the Punnett scale. So we will do self-fertilization. So this is the one side, and this is the other side. After fertilization, at each scale, the phenotype and genotype will be like this. Finally, the ratio will be 9 to 3 to 3 to 1. So even though Mendel did not know that do allele encoding different traits separate independent. He didn't know that, but he found that the ratio is always 9 to 3 to 3 to 1. Based on his observation, so this result only is comes from the independent assessment. So the allele encoding color separate independently of allele encoding seed shape. Only in this case, the result will be the 9331. Okay. How about we will see this principle in the chromosome at chromosome level. In this slide, I will explain the dihybrid cross based on the meiosis. So the principle of independent assessment is highly related to the meiosis. Okay. What was the principle of independent assessment? The principle of independent assessment means that allele encoding one phenotype is independent, separate independently of allele encoding another phenotype. But this one is very hard to understand. But if you relate the principle of independent assessment to meiosis, it will be very simply can be explained. Okay, so the F1 generation will be the large R, large Y, and small y. So because the genes encoding the shape and genes encoding color is located at different chromosomes, chromosome 1 and chromosome 2. So during the meiosis 1 interphase, the DNA will be replicated like this. Okay, at that, during the metaphase 1, each chromosome pair, homologous pair is aligned together. This is metaphase plate like this. They are aligned together. At that, during anaphase 1, this part will go to the one part, and this part will go to the another pole. So they will make this daughter cell. They will make this daughter cell. After meiosis 2, they will make the gamete or large R, or large R, large Y, large R, large Y, or small r, small r, or small y, small r, small y, like this. How about when they are doing the metaphase 1, when they align together? How about the small r, small y can go to the in this location. At this case, at anaphase 1, one daughter cell will have the large R. One daughter cell have the small r, small y, and small y. And one daughter cell, they will have small r, and large y. And they will finally, after meiosis 2, they will have this gamete like this. So if the genes located on different chromosomes, for example, genes encoding the shape and genes encoding color is located at different chromosomes, and also the genes will also independently based on the meiosis. So if we relate the principle of the independent assessment to meiosis, we can easily explain the principle of independent assessment. In this slide, I will explain the branch diagram. Okay, in dihybrid crosses, is very difficult to predict the expected offspring. For example, this is dihybrid crosses, After that, the phenotype ratio is 9 is the round and yellow, 3 is round and green, 3 is wrinkled and yellow, 1 is wrinkled and green. So this is very difficult to calculate, but if you use the branch diagram, it's very easy to determine the phenotype and the expected proportion of offspring from a dihybrid crosses. Okay, let's start from here. Okay, in F1 generation, we will have the large R, large Y, small y. So we will do the self-fertilization. Okay, so we will do self-fertilization. So at this cases, the branch diagram is the broken. Dihybrid, dihybrid cross can be broken into two monohybrid crosses. Okay, just the broken here and broken here. So we broken here and we broken here. So one is here and one is Y is like this. So based on the large R, small r, and cross with large R, small r, so expected phenotype will be the wrinkled will be the 1 out of 4, and the round will be 3 out of 4. How about the color? Color will be the 1 out of 4 will be the small y will be the green, and also yellow will be the 3 out of 4. Okay. So and now we will calculate the expected proportion for both character. Okay. At this cases, this calculation can go to the here, and also this calculation can go to the here and here. Okay. After that, we can predict it like this. Based on this one, round and yellow will be the 3 out of 4 multiply 3 out of 4 will be the 9 out of 16, like this. How about this one? Round and green. Round and green means that 3 out of 4 multiply 1 out of 4 will be the 3 out of 16. So round and green will be, the proportion will be like this. How about this one? Wrinkled and yellow. Wrinkled and yellow will be the 1 out of 4 multiply 3 out of 4 will be the 3 out of 16. So this is the wrinkled and yellow ratio. How about wrinkled and green will be the 1 out of 4 and 1 out of 4 will be the 1 out of 16. So finally the ratio will be 9 to 3, 3, 1. So if we use the branch diagram, it will be very easy to determine the phenotype and also determine the expected proportion of offspring from a dihybrid crosses. Just divided by 2, broken. Dihybrid crosses can be broken into 2 monohybrid crosses. After that, we can combine to each other like this. Okay. In this slide, we will cover dihybrid test cross. Previously, we covered monohybrid test crosses. It is nearly similar to each other, but the difference is that the dihybrid test cross is the one individuals of unknown genotype, one individual of unknown, but they have 2 alleles, which encode the shape and color, crossed with another individual with homozygous recessive genotype. Homozygous recessive genotype means that wrinkled and green, small rr, small yy. So if one individual of unknown genotype is heterozygous like this, or they can be homozygous, large R, large R, large Y. But at this case, when they are in the heterozygous like this, the expected proportion per post character will be, if we can split it like this and split it like this. The expected proportion per post character will be the 2 out of 2. 1 out of 2 is round. 1 out of 2 will be wrinkled. How about in these cases? The expected proportion per second character will be the 1 out of 2 will be the yellow, and 1 out of 2 will be the green. And the expected proportion per both character, we can combine to each other. This can go to the here, and this can go to the here and there. At that, we can do multiply to each other, and round and yellow will be 1 out of 4, and the round and green will be 1 out of 4, and wrinkled and yellow will be the 1 out of 4, and wrinkled and green will be 1 out of 4. In this slide, when we cover when one individual's unknown genotype is large R, large Y, large Y. The phenotype is round in shape. When this case is, the expected proportion for post character will be the 2 out of 2 will be the round. How about the second character? Second character is also 2 out of 2 will be the yellow. So how about the expected proportion for both character? So this one can go to the here, and this one can go to the here. So after multiplication, all of them will be the round and yellow. So okay, dihybrid test crosses tell us if the ratio is 1 to 1 to 1 to 1. In this case, this genotype will be the heterozygous, or all of the phenotype is the 1. At this case, we can expect that the genotype of one individuals of unknown genotype will be the homozygous and dominant, large R, large Y, and large Y like this. So dihybrid test crosses tell us whether this is the homozygous or whether this is heterozygous. Okay. So in this slide, we will cover the final subject. The observed ratio of progeny may deviate from the expected ratio by chance. It's a little difficult to understand, so I will explain it very detailedly, okay? So, for example, this one is for example, this is dihybrid test cross. The expected value when this case is happened, the expected value will be the 1 to 1 to 1 to 1. But this is our expectation. But observed value will be different. Maybe it's different. We don't know. Who knows? So, okay. So at this case we will do the test chi-square goodness of fit. Okay. First, to calculate the chi-square goodness of fit, we have to make the null hypothesis. Null hypothesis. In Korean, we call it as 기무가설. 기무가설 means that assume that there is no significant difference between the observed and expected value. So this is the calculation, the calculation of chi-square goodness of fit. So this is the observed value minus expected value and multiply that divided by expected value. And also we have to know the degree of freedom. Degree of freedom is n minus 1. What is n? N is number of different expected phenotype. Okay. in this case is how many number of expected phenotype. Number is 1, 2, 3, 4. In this case is what is the degree of freedom? 4 minus 1 will be 3. Okay. Next, next I will explain the procedure to calculate the chi-square goodness of fit. Okay. First, find the rho for the appropriate degree of freedom. So, in this case, what is a degree of freedom? 4, 1, 2, 3, 4 minus 1 will be 3, isn't it? So at this case the degree of freedom will be 3. This is the degree of freedom will be 3. Okay. After that, define where the calculated chi-square value located. Okay. Where, so we define the degree of freedom. After that, we define where this number is located. Here and there, and here and there, where is it located. After that, we can define the where the probability of chance located again. After that, if the probability of chance is greater than or equal to 0.05, for example, if the p-value is greater than 0.05, it means that there is no significant difference exists between observed and expected value. When the probability of chance is greater than the 0.05, it means that significant difference existed between observed and expected value. So a little bit difficult to understand. Okay, so at this case chance is responsible for difference. And in this case, chance is not responsible for difference. Okay, let's solve one problem. This slide, we will solve one chi-square goodness of fit example because this is very difficult to understand. Okay, let's start. In parent generation, we crossed homozygous. We crossed the purple flower, which has homozygous dominant genotype, and also cross with the white flower, which have the small pp. The large p is dominant than small p. So F1 generation is all of them is purples. So after that, we did self-fertilization. So based on the Punnett scale, okay, so the expected value is 3, 1, 2, 3 out of 4 will be 3 out of 4 will be purple, and 1 out of 4 will be white. This is our expected value, but we observe that, and after fertilization, we observe that 105 showed the purple color and 45 showed the white color. So this is the observed value. So 105, 45 total is 150. So our expected value is 3 out of 4 multiply 150 is the 112.5 is our expected value, and also 37.5 is our expected value. So now we will calculate the chi-square goodness of fit. So this is chi-square goodness of fit. So one is the observed value is 105, and expected value is 112.5, and multiply and divided by 112.5. The other one is that observed value is 45 minus 37.5. After that, multiply and divided by 37.5. After that, this value, after we calculated, the value is 2.0. Okay. After that, so defined. after that we have to find the law for the degree of freedom. So we calculated the chi-square goodness of fit. Okay. After that we have to find the degree of freedom. So what is degree of freedom? So what is number of phenotype? Phenotype is purple or white. It will be 2. 2 minus 1 will be 1. Okay. Next, what we have to do? So we have to define where the calculated chi-square value located. Okay. So we will see that we calculated 2.0. So we have to know where this value is located. Okay. So the degree of freedom is 1, okay? So 2 is located between 0.455 and also 2.706, like this, located here. So it means that the probability chance is located between 0.1 and 0.5, okay? And it means that the probability of chance is greater than 0.05. So the value is that the value is between the here between the less than 0.5 and the larger than 0.1. This value is larger than greater than 0.05. It means that there is no significant difference exists between the observed and expected value. So this is the procedure how to calculate the chi-square goodness of fit. Okay, once again, first we have to find the law for the degree of freedom. After that, calculated chi-square goodness of fit based on the observed value and expected value. After that, we can define where the chance is located. If the probability of chance is greater than the 0.05, it means that there is no significant difference. But chance is less than 0.05, it means that there is a significant difference between the observed and expected value. Okay. 3, we cover the basic principle of heredity. It was very difficult, isn't it? The first, we cover the Mendel, and also second, we cover monohybrid crosses, and we also cover dihybrid crosses. Finally, we know the chi-square. We know how to calculate chi-square goodness of fit. Okay. Okay, today we will finish our class. Okay.